So chapter 13 is hair and fibers. So here we go. So it's trace evidence. It's all about trace evidence. So chapter 13 and 14 are about trace evidence, which includes hair, fibers, soil. Okay, so if we start here, hair is physical evidence in a lot of crimes. So it's everywhere. Okay, now it's not possible yet to individualize a human hair based on the shape of it, so based on the morphology of it. However, if there are cells present, then we can look at the DNA and individualize it that way. Okay, so you can't do it with the shape of the hair, but you could do it if there's DNA. Okay, so when properly collected and submitted to the lab with an adequate number of reference samples, hair can definitely provide corroborative evidence for placing an individual at a crime scene. So hair is used all the time to demonstrate that someone is present. Okay, so if we talk about the morphology of hair, so what is hair? So it's an appendage of the skin. It grows out of a hair follicle. Okay, the length of the hair extends from its root or the bulb that's embedded in the follicle, in the hair follicle, and then it continues into what's called a shaft, and then it terminates at the tip. Most of the information that we get from hair is obtained from the shaft of the hair. So the shaft has 3 layers. It has a cuticle, which is the outside layer, the cortex, and then the middle layer, which is called the medulla. Okay, so the cuticle is this, it's kind of like a scale, and it covers the outside. So the scales always point towards the tip, so you can orient the hair, because the scales should always point towards the tip. Okay, so the scales kind of maybe look like scales on a fish. Okay, but they're pointing towards the tip instead of towards the tail. Okay, and then the scales can be different shapes. So if the scales are different shapes, you can use that to identify what species of animal the hair comes from. So is it from a dog? Is it from a deer? So that's useful. Okay, now the cortex is the main body of the hair. So it's important because that's where the pigment or the color is. So the pigment granules that make our hair different colors are found in the cortex of the hair. So that the pigment granules could be different colors, they could be different shapes. So it gives the criminalist or the lab technician something to compare. So you can use them, you can compare the pigment granules. So they're like little, maybe like salt granules in there, but they're different colors. Okay, so the book has a picture of skin, so it's a cross-section through skin, and it shows the hair growing out of the follicle. So the follicle is kind of like a tube, and the tube goes down into the dermis, and then the hair grows out of there. There are nerves around it, so when you pull your hair, you can feel what's going on. Okay. There's another picture from the book, and it shows different kinds of scale patterns. So there's one called coronal, and coronal kind of looks like a crown. So, or you could think of it as a stack of paper cups. Okay, so the cups are all kind of stacked together. Sometimes there are little like arches on the cups too. So the cups maybe have been cut to make them different shapes instead of just being totally round at the top. Okay, we also have scales that can be spiny or shaped kind of like petals on a flower. Okay, so the tips are always going to be pointed towards the end. And then there's another one, it's called imbricate or flattened scale. So those are kind of funky. It's more like a puzzle, I think, where it's a long hair, and then it's almost like maybe an irregular lattice. So a lattice would be very regular. Everything would be kind of equally spaced and distant apart. And this is more like a bunch of lines that are, I mean, they're meeting from side to side, but they're not in a regular pattern. They're irregularly patterned. Okay, now the medulla is the middle that's running through the center of the hair. So it's got cells in there. Well, no, it's not cells, but it's the column where the cells would have been. Okay, so the medullary index is going to measure the diameter of the medulla, and what the index does is it compares the diameter of the medulla to the diameter of the hair shaft. Okay, so you're comparing the two. So how wide is the medulla compared to how wide is the hair? So in humans, the medulla usually is less than 1/3 the diameter of the shaft. In animals, it's bigger. It's usually 1/2 or greater. So animals have a bigger medulla than humans do, typically. Okay, now the medulla could be continuous, so it goes all the way from like the bulb all the way up, or it could be interrupted, so it's kind of maybe like the line has spaces in between it, or it could be fragmented. So fragmented means it's kind of like it's broken up, it's not in a regular pattern, or maybe they don't have a medulla. The presence of the medulla varies between individuals and even in one individual's hairs. So there's lots of variation. So if you take the 25 hairs, they might not all look the same. So the middle of it, the medulla might not be the same. So, and depending on what species of hair, the hair comes from, the medulla could have different shapes. Okay, so they're not always consistent. Okay, now if we look at the root of the hair, so the root and the other surrounding cells in the hair follicle, those are where the hair is gonna grow from. Okay, so they're gonna be down in the in the dermis of the skin. Okay, now hair has 3 growth phases. So anagen, catagen, and telogen. So anagen, trying to find it here in the book. So in the book, it's on page 323. So anagen is when it's first growing. Okay, so the hair the hair follicle is starting to produce hair. The catagen is in between, it's in the middle, and then the telogen is when the hair actually falls out. Okay, so your hair starts out in anagen, so it's like ACT, so, or AC&T, which kind of helps because we have that company. So anagen is first when it's first starting to grow, catagen is in the middle, and telogen is at the end when it actually falls out of your head or your skin. Okay, so if hair is being pulled from your head, some tissue may be around the hair shaft and near the root, okay, and that's called a follicular tag. A follicular tag is someplace where we can get DNA. So you can get DNA out of there, and then you could individualize, you could figure out who the hair actually belongs to. Here are some pictures where you can see, well, yeah, I don't know, I guess you can see. So there's anagen where the hair is first starting to grow, so the root is kind of big. Catagen, the root is kind of, it's more condensed, I guess. And then the telogen phase, it's the tag, it looks like the cells are dying. Okay, it looks like it's dead. So if you were trying to get DNA, you probably want to go for anagen or catagen, because telogen, if the hair is falling out, it's not going to have living cells on it. Okay, so a comparison microscope is what's going to be used to look at hairs. So we can compare the scales. You can look to see if there's a medulla. You could figure out the medullary index, so you could compare the size of the medulla to the size of the hair. You could look at the shape of the medulla, and you could look at the distribution, the shape, and how intense the colors are in the pigment granules. Okay, so all those things are gonna make the hairs look different, hopefully. Okay, so the most common request that the criminalists get is the hair from a scene from a suspect. Okay, so is this hair actually from somebody? So sometimes it's hard to say. Okay, so the hair examinations that are done with a microscope are very subjective, and they depend on the skills and the integrity of the person doing the test. So the best way to do it would be with quantitative data. So reported hair comparison must contain DNA information. So that's going to give you more. It's quantitative, it's a number, or it's a percentage that you can say, yes, this is, there's 1 in a billion chance that these markers match with DNA. But with hair, it's more, what does it look like? So it's more qualitative. It's harder to do and harder to use. Okay, so now another question on here is, are efforts being made to individualize human hair? So, I mean, we have to use DNA to do that. So right now, that's where the push is. So they're trying to get better at getting DNA from hair. And then, is it possible to determine whether hair came from a deceased individual? So they say maybe, right? But it's the hair, if someone's dead, depending on how long they're dead, some of the hair has begun to decay or break down. So it is possible to determine whether hair came from a deceased person, possibly, maybe. Okay, so now another question on here is, are efforts being made to individualize human hair? So, I mean, we have to use DNA to do that. So right now, that's where the push is. So they're trying to get better at getting DNA from hair. And then, is it possible to determine whether hair came from a deceased individual? So they say maybe, right? But it's the hair, if someone's dead, depending on how long they're dead, some of the hair has begun to decay or break down. So it is possible to determine whether hair came from a deceased person, possibly, maybe. Okay, so then hair, so hair and DNA. So if you have cells and you can get the nucleus out of the cell, then you can use PCR and you could get DNA. It's possible. The probability of detecting DNA in hair roots is more likely for hair that's in anagen, so that's the early phase, compared to catagen, which is the middle, remember, or the telogen phase. So anagen's where you're most likely to get hair. If hair is forcibly removed lots of times, then the follicular tag is there, and that is a place where we can get DNA out of. You can get mitochondrial DNA, so there are these other organelles in your cells that are mitochondria, so you can get mitochondrial DNA from the shaft of the hair, but that will just, that's not, you can't individualize someone, that's more class. So it's makes it easy to, or not easy, but you could track somebody to their mom, but you couldn't necessarily track, you could say, okay, these people had the same mom, but you might not be able to say for sure who the specific person is. Okay, so mitochondrial DNA comes from mitochondria, and they're in the cytoplasm of the cell. So it's transmitted mom to dad. It can't be used to individualize a hair, but if you have morphological data, so if you could look at the hair and you have other data, it could help provide more pieces of a puzzle. So it usually, if you're doing hair comparisons, you have to also have some DNA from them. Okay, so now in general, forensic hair comparisons involve head hair or pubic hair. So to collect hair as a reference, you need to have 25 full-length hairs from all areas of the scalp. And then this is where I have a mistake. So a minimum collection of 25, and I have dozen in here, full-length pubic hairs, but it's just 25 pubic hairs too. So their hair samples are collected if during an autopsy from victims of suspicious death. So that's just kind of a routine thing that they do. Okay, now fibers. Natural fibers come from animal or plant sources. So examples of fibers that come from those that are natural are like wool, mohair, cashmere, fur, cotton, linen. Okay, so man-made fibers are manufactured. So usually there are two categories of those. So regenerated fibers are made from natural raw materials, and those include rayon, acetate, and triacetate, which those two you might not see very often. And then synthetic fibers are produced from chemicals, so things like nylon, polyester, and acrylic. Okay, so fiber evidence, it depends, the quality of the fiber evidence depends on the ability of the criminalist to identify the origin of the fiber or to narrow the possibilities to a limited number of sources. So if the examiner has fiber that's torn and they could match the edges, then they could definitely confirm that the fabrics were from the same place. Okay, so microscopic comparisons between questioned and standard reference fibers are usually compared for color and diameter, and they use a comparison microscope. Okay, now other characteristics that are important when you characterize fibers or compare fibers, lengthwise striations, delustering particles that reduce shine, the cross-section shape of the fiber, so whether it's flat or triangular, and then differences between dyes. So compositional differences may exist in the dyes that were applied to the fibers during the manufacturing processes. So lots of times fibers have a dye lot, and the dye lots can vary depending on who's making them. Okay, so here's cross-sectional shapes of fibers. So they could be round, they could be flat, they could be multi-lobed. So that kind of looks like a star or Patrick starfish, right, on SpongeBob. They could be trilobal, so that means they're kind of almost like a triangle, or shaped like a dumbbell. Now, people spend lots of time, they look at wools, they look at all sorts of different fibers, natural fibers, to compare them. So there are definitely experts in fiber shapes. Okay, now infrared spectrophotometry is a way to identify generic classes of fibers. So the molecules that make up manufactured fiber absorb infrared light differently. So you can compare the way that they absorb the light to determine what they are. So this thing, it's called a microspectrophotometer. So it's a combination of a light microscope and a spectrophotometer. And what it does is it allows you to view an object under the microscope while obtaining its infrared absorption pattern. So it's a really nifty way to compare fibers. It's really expensive. It's a very, very specialized piece of equipment. Okay, now the investigator, they need to look for minute fibers. They often, okay, so the task of looking for fibers often becomes one of identifying and preserving potential carriers of fiber evidence. So they have to collect different articles of clothing, and they should be packaged in separate bags. If it's necessary to remove a fiber, they should use clean forceps, place it in a small sheet of paper and fold and label it, and then place that little paper packet that you've made inside another container. There's an introduction to the chapter. It's about fatal vision. Fatal vision was about, it happened at Fort Bragg. Captain Jeffrey McDonald, he bludgeoned his wife and he killed his kids. And how they actually found it was because he had his pajama top was stabbed with an ice pick, so with the weapon. And they could tell how the fibers were stabbed, that it was self-inflicted. Actually, he did to the pajama top when it was on the ground. And then they also found the pajamas in everybody's room, and they were with the bodies. So he was kind of convicted because of his fiber evidence. And that was in the 70s. Okay.